Okay. Thanks so much for Professor Gawar to have me. And I was really expecting to have more time for my talk, than 20 minutes, but then I will be very fast. So excuse me about this. So today I will talk to you about biodiversity genomics in the omics era. And my talk layout will be a brief introduction about the biodiversity genomics field, and then genomics techniques, and a brief glance in the Earth Biogenome project, and then we will have a glance in the future outlook. First of all, I forget to introduce myself. I'm Abdollah SHARAF. I'm currently senior bioinformatician in Sequenas, the sequencing facility in University of Constance, Germany. So for introduction, biodiversity genomics and also is known as a biogenome. It is a fast and relatively new field to study the biological variation of organisms in the layout of its DNA. So by definition, it's coming from the terminology, so bio and genomics, then it is about genomics and biodiversity. And for sure, as all of you know, genomics meaning to study the structure, function, and evolution of organism entire genome. And biodiversity, for sure, you know what is biodiversity. It is a variety of life Earth. Importance, for sure, really it has so many importance, but just I will briefly go through some of them. So for sure, to study the genetic diversity of species, identify, classify species, and even reconstruct the evolutionary relationships. For sure, investigates genetic basis of adaptations. This is actually what I think Gawar mentioned in his first talk. And for sure, provide insights about the ecological function of different organisms within environment, or known with the environment-organism interaction. And also to study the effect of the toxic creature. We talk about human here. To habitat destruction, climate change, of course, and pollution, etc. Also to understand the complex interaction between organisms and their environment. And for sure, many more other application importance. For application, for sure, is coming from the fact we can have species identification, phylogenetics, study, drug discovery, conservation, of course, but actually my favorite application, what is known by bioprospecting. And bioprospecting, it is searching for and discovering new sources of potential use for biomolecules or natural resources. And for sure, all of those molecules, we can find it in the highly biodiversity region like Algeria, for sure. And for sure, all what I talk, all this genomic biodiversity, it was not possible without the genomics techniques and the development of this field. And omics as a technology, as a terminology, it is about all the techniques ending with omics. And for sure, in the genomics biodiversity, we use most of those techniques, but we mainly focus on 4 of them: DNA sequencing, metagenomics, transcriptomics, and even proteomics. But actually, all of these genomics projects start with the DNA barcoding. I don't know how many of you familiar with DNA barcoding, but I know it's really well known now. It is a technique to identify species by analyzing a short or now the standard segment of their DNA. And it is really very simple technique, including PCR. And as you know, PCR, we needed to amplify a certain segment in DNA. And now it's really very standardized. So now we know which region to amplify, and even for which organism or group of organisms. And this region now is known as a barcode regions. So now I will introduce about like one of the biggest initiative based on this DNA barcoding, which is barcode of life. I think most of you already use it in their research, I think. And barcode of life, it is an initiative to create library of DNA sequences from all living Earth organisms. And actually it's the goal, but it's not the goal now. Actually, they already achieve it, developing or when they assemble the whole human genome. And then, so these mean you have to go with these techniques, with these gel-based techniques, to have 3 million bases there to have the human genome. Then it was really normal to see such massive labs. This picture from the Craig Venter sequencing lab, actually. And I think this is one of several, simply only to just like having like 3 million bases. Then it was logically to searching for new, more efficient techniques, sequencing techniques. Because of this, we switched to the next or second generation one, which is mainly about trying to solving the defect of single sequencing, meaning having more sequencing capacity and like parallel one. And this technique is based on the cutting your DNA of the whole entire genome of organism, and then apply it in some smart chip. And again, as I told you, this technique still is sequencing by synthesis, and then we detect the nucleotide. And actually, this really, again, it was really, by this time, it was really evolutionary, because you can see this really how this affects the cost of a megabit. So you can see really once this technology start, you can see the prices really dramatically drop. But it's still like really all techniques coming with disadvantage. So since we start with cutting our DNA into pieces, then we need afterward to assemble. And also even some initiative, European initiative, now they implement the AI to identify species from sounds, from bird sounds. And for sure also with the functional genomics, so like artificial intelligence and functional genomics start as really totally different field, but then by millennium is already start to overlap. And actually if you go and look to the implementation of AI, you can see it is already implemented in all the omics one. And for sure, even now with functional genomics and related to the biodiversity genomics, it is already well implemented in the gene prediction, because one of the main tools we use for gene prediction for our genome, it is this AB initio prediction. And for sure, these extend to the 3D structure of the protein. And for sure, all of you heard about AlphaFold and AlphaFold 2 announcement. Last but not the least, actually, even also the future outlook, and actually this one of the initiative coming from Africa BB about the genomics biodiversity outputs. Because for us, the idea not really sequence genome, but also how to deliver the knowledge to all communities within Africa. So now also we study how to implement MI to have to translate the research outcomes of the genomics biodiversity into the local languages. And that's it. Did I made it in 20 minutes? No.
20 minutes.
Okay.
Thank you. It's very good. Thank you, Dr. Abdullah.